I will start now with the general information for the participants, and since they're coming in, and I just wanted to say hello again to everybody and start thanking our speakers today, Professor Deswan, Professor Jauerbaum. And also I wanted to remind you all that we have the possibility of writing a review or original article for 3 or 4 different journals associated with the course. And the idea is that you contact the speakers and try to produce a manuscript together. So that is because one of the aim of the course is to increase the possibilities for students mainly, and also because we have such good speakers in the course, it is an honor to write something together with them. So Gernot and Alicia will be also available for that. So try to take this option, write to me to put in the list of potential contributors to these reviews and original articles in different journals. So we will start now with the section number 6 of the course, and I have the pleasure of introducing 2 very well-known speakers. First is Professor Alicia Jauerbaum from the University of Buenos Aires in Argentina. Then the other one is Professor Gernot Deswan at the Medical University of Graz in Austria. The first speaker this morning or this afternoon, and in some regions this evening, is Professor Alicia Jauerbaum. Thank you very much, Alicia, for being here. She's director of the Laboratory of Reproduction and Metabolism at the University of Buenos Aires, and principal investigator for CONICET in Argentina. It's a very important entity in Argentina supporting the research for scientists. She has been president of the Latin American Society for Placenta and Maternal Fetal Interaction, SLIM, you may know that, and is a board member of the International Association of Diabetes and Pregnancy Study Groups. She has many prizes from different entities, and is, of course, she received a prize from the Sociedad Argentina of Diabetes award in 2020, and many others. So we are very happy that we have today the pleasure of listening to Alicia. So please go ahead.
Thank you very much, Luis, for the introduction, and I'm very happy to participate in this course. And I will talk about fetal programming in maternal diabetes. And first, I would like to start with diabetes, a metabolic pathology, very prevalent, diagnosed by hyperglycemia. And in this is a pathology characterized by metabolic alterations in carbohydrates, proteins, and lipid levels caused by an insufficiency in the secretion and or in the action of insulin. And we can distinguish the main forms of diabetes: type 1 diabetes, resulting from a specific autoimmune aggression to the beta cells, the producers of insulin. Type 2 diabetes, resulting from mechanisms of insulin resistance associated with lack of appropriate pancreatic response. And gestational diabetes mellitus, which initiates during pregnancy, mostly due to lack of adaptation to the metabolic changes that occur during pregnancy. And I will talk today about maternal diabetes, and this is important as it increases risks of several reproductive anomalies. Indeed, there are increased risks of spontaneous abortions and congenital malformations induced by pregestational diabetes, both type 1 and type 2 pregestational diabetes. And there are increased risks of fetal impairments, placental alterations, perinatal morbidity and mortality, and developmental programming of metabolic and cardiovascular diseases induced by both pregestational and gestational diabetes. And this is important as we are talking about prevalent pathology. 1 on 6 to 10 pregnant women has any form of maternal diabetes during their pregnancies. Now, experimental models are needed to understand the physiopathological processes in diabetic pregnancies, and in our lab we have characterized a mild diabetic rat model induced by the neonatal administration of streptozotocin. This drug distracts the beta cells in the neonatal period, followed by partial regeneration of the beta cell mass, leading in the adult animals to a model characterized by moderate hypoglycemia, moderate hypoinsulinemia, and compatible with the pregnant state, and useful to study the offspring. Indeed, the offspring at 2 days of age, the animals show normal circulating levels of triglycerides and glucose. But from day 14 of age, they show increased levels of circulating triglycerides. And from the 5th month of age, these animals show hyperglycemia. Therefore, studying these models, we learned that in the offspring, although lipid concentrations precede glucose metabolism improvements, and that gestational diabetes programs type 2 diabetes in the offspring. Indeed, we started with the mother with mild diabetes, in which the mother has hyperglycemia due to the low insulinemia, but in which the fetuses show hyperglycemia and hyperinsulinemia, and we obtained 5-month adult offspring with hyperglycemia and hyperinsulinemia. Moreover, the 3-month-old adult offspring is normoglycemic, but if we mate this offspring with healthy males, a GDM model is obtained characterized by hyperglycemia and hyperinsulinemia at their term pregnancies. Therefore, we have developed animal models to study pregestational diabetes, the offspring previous to diabetes development, type 2 diabetic rats, and GDM rats generated through intrauterine programming. On the other hand, our group has contributed to understand that the pro-oxidant and pro-inflammatory environment characterize the intrauterine compartment in both experimental models of diabetes and in diabetic pregnant women. And we focused in the study of the nuclear receptor PPARs, addressing if there are important PPAR pathways related to this adverse intrauterine environment. The PPARs are ligand-activated transcription factors that regulate the expression of target genes involved in developmental processes, metabolic homeostasis, and antioxidant anti-inflammatory pathways. There are 3 PPAR isotypes, PPAR alpha, PPAR delta, and PPAR gamma. The 3 of them are activated by ligands of lipid nature. Indeed, different polyunsaturated fatty acids and monounsaturated fatty acids can activate the 3 PPAR isotypes. There are different arachidonic acid derivatives that activate each different isotype, and there are different pharmacological agents that activate each isotype, as fibrates, which are hypolipidemic drugs that activate PPAR alpha, or glitazones, insulin sensitizing drugs that activate PPAR gamma. Now, it is interesting to think as nutrients as agents capable of regulating transcription. Indeed, PUFAs and MUFAs are nutrigenomic agents capable of activating the nuclear receptor PPARs and leading to the regulation of gene transcription. Now, I don't have time today to show you our results addressing that maternal diabetes leads to impairments in PPAR levels and PPAR function in rat embryos organogenesis, in different fetal organs, and in the placenta of diabetic rats and of women with GDM. And besides, we found that diets-enriching MUFAs and PUFAs prevent embryonic, fetal, and placental impairments in these experimental models of diabetes. And I will like to show you today our results addressing the effects of maternal diets enriching PUFAs or MUFAs in the offspring of these diabetic animals and in the placenta from GDM patients. So in these studies, we hypothesized that the diets enriching MUFAs and PUFAs, which are efficiently transferred from maternal circulation through the placenta to the fetus and can activate nuclear receptor PPARs in the placenta and the fetuses, induce changes with putative beneficial effects in the offspring. So in these studies, we treated the control and diabetic animals with diets enriching MUFAs or PUFAs in the context of a normolipidic diet from day 1 of pregnancy and until birth. We stopped the treatments at birth and studied the offspring. So in these first studies, I will share with you some results. We found addressing the effects of a PUFAs enriched diet throughout the FO pregnancy in this model of pre-gestational mild diabetes, and studied their effect in the 3-month-old F1 offspring, the animals that develop GDM. So in this experimental design, we start with the healthy and mild diabetic animals, and during the FO pregnancy, the animals were fed with diets enriched or not with safflower oil from days 1 to 14 of pregnancy to provide diets enriched in linoleic acid and N6-PUFA highly needed during organogenesis, followed by a diet enriched in 6% chia oil, highly enriched in linoleic acid and N3-PUFA. And these treatments stopped at birth and no dietary treatments in the offspring. At 3 months of age, the offspring was mated. This is the F1 pregnancy in which GDM is expected to develop, and we studied in animals on day 21 of pregnancy. And we found that the offspring of these mild diabetic animals at 3 months of age still develop GDM. So the animals that received the diet enriched in PUFA in the FO pregnancies show higher hypoglycemia, similar to the ones that received the standard diet. But these diets were able to prevent hypertriglyceridemia and hypercholesterolemia in the GDM animals. We also observed increased lipoperoxidation in the placenta, and the capacity of these diets enriched in PUFAs in the FO pregnancies to prevent desalteration. And we think that this is probably related to the capacity to regulate pipogamma levels, which are reduced in the GDM placentas and prevented in the placentas of the GDM animals whose mothers received the PUFAs enriched diet in the FO generation. Besides, we found that GDM leads to fetal overgrowth and that the diets enriching PUFAs in the FO generation prevent desalteration. There are no changes in placental weight in this experimental model. And as fetal overgrowth is highly related with mTOR pathways activity in the placenta, together with the group, in collaboration with the group led by Thomas Johnson and Teresa Powell, we addressed placental mTOR pathways, finding that mTOR1 pathway is overactivated and desalteration prevented by the maternal diet enriched PUFAs in the FO generation. Therefore, PUFA supplementation in the FO generation prevents hypertriglyceridemia, fetal overgrowth, and placenta hyperoxia and pro-inflammatory markers in GDM rats, at least in part through the regulation of PPAR and mTOR pathways. We next focus on diets enriched in extra virgin olive oil, as they are composed mostly of oleic acid and monounsaturated fatty acid, and also N6 polyunsaturated fatty acids, nutrigenomic agents capable of activating the nuclear receptor PPARs, and also have minor components that are relevant, such as polyphenols, due to their antioxidant properties. And there are several relevant studies addressing the potent effects of extra virgin olive oil supplementation in the prevention of cardiovascular diseases and in the anti-inflammatory effects in other pathologies, but little is known about the putative effect during gestation and in the offspring. And in these experiments, we treated with the 6% olive oil enriched diet during the FO pregnancy and studied the offspring. We first studied the prepubertal offspring and found we focused in the heart of this offspring, addressing that there are increased lipoperoxidase, increased peroxynitrite-induced damage, indicating a peroxidant and pro-inflammatory environment that was prevented by the maternal diet enriched in olive oil. There were also increases in connective tissue growth factor, a profibrotic and pro-inflammatory marker that was reduced in the heart of these diabetic offspring when their mothers were treated with the diet enriched in olive oil. And we also found increased apoptosis in the heart of this diabetic offspring, an alteration prevented by the maternal diet enriched in olive oil. Therefore, maternal diets enriched in olive oil protect the heart of the 21-day-old offspring from the peroxidant and pro-inflammatory damage programmed by maternal diabetes. We address also the 5-month-old offspring of these diabetic animals, the animals that develop type 2 diabetes. And we found that the heart of these animals accumulate lipids, phospholipids, cholesterol in females, triglycerides in males, alterations prevented by the maternal diet enriched in olive oil. And there were also increased lipoperoxidation and nitric oxide production, alterations prevented by the maternal diet enriched in olive oil. Together with the group led by Edith Allan in Canada, we studied endocrine pancreas in the male offspring of these diabetic animals, finding reduced beta cell mass, islet area, and number of large, small, and total islets in the offspring of these diabetic animals, and that the maternal diet enriched olive oil prevent these alterations. Beta cell clusters were also altered in these animals, an alteration prevented by the maternal diet enriched in olive oil, and there were also changes in PPAR's expressions, no changes in the expression of PPAR alpha, but reduction in the expression of PPAR delta and PPAR gamma in the endocrine pancreas, and the expression of PPAR delta was prevented by the maternal diet enriched in olive oil. Therefore, alterations in the heart and endocrine pancreas are prevented in the adult offspring of diabetic rats treated in the F0 generation with an olive oil enriched diet. Now, what happens if we treat the F1 pregnancies, the pregnancy that develops GDM? So in this different experimental design, there were not, we started again with the healthy and mild diabetic rats, but there were no treatments in the F0 pregnancy. The offspring was mated at 3 months of age, and the F1 pregnancy was, at F1 pregnancy, the animals were treated with 6% olive oil enriched diet. And we found that maternal glycemia is still elevated. These animals treated with olive oil still developed GDM, but maternal triglyceridemia was prevented. We focus in the liver as a target organ in the fetuses, finding that the liver of male fetuses accumulate lipids, triglycerides, and cholesterol alterations prevented by the maternal diet enriched in olive oil. And this was related to the increased levels of perilipin-2, a protein involved in lipid droplet formation and stabilization, increasing the GDM group, an alteration prevented by the maternal diet enriched in olive oil. Now, PLIN2 is a target gene of PPAR gamma, and PPAR gamma was also found increase in the livers of male fetuses in the GDM group, an alteration prevented by the maternal diet enriched in olive oil. We next focus in microRNA 122 in the fetal livers. As this microRNA is the most abundant microRNA in the liver, it is associated with the hepatocyte differentiation, lipid metabolism and function, and its relation-induced hepatic steatosis and fibrosis. And it is also a negative regulator of the nuclear receptor PPAR delta. And we found reduced levels of MIR122 in the fetal liver of these male fetuses in the GDM group, an alteration prevented by the maternal diet enriched in olive oil. And this occurs in parallel with an increase in people delta levels in the GDM group, and alteration prevented by the maternal diet enriched in olive oil. Therefore, a maternal diet enriched in olive oil prevents lipid overaccumulation, alters PPAR pathways, and reduces MIR122 expression in the liver of male fetuses of GDM rats. Now, we focus also in the placenta and found that the PPAR gamma levels reduced in the placentas from the GDM group are still reduced when the mothers were treated with the olive oil enriched diet in the F1 pregnancies. But we observed antioxidant and anti-inflammatory effects. Lipoperoxidation increase in the GDM placentas was prevented by the maternal diet enriched in olive oil. There were also increases in CTGF and matrix metalloproteinase 2 levels prevented by the maternal diet enriched in olive oil. And therefore, with these studies addressing diet supplemented with olive oil in the F1 pregnant rats that developed GDM, we found that GDM is not prevented by maternal hypertriglyceridemia, is prevented. This treatment regulates PPAR levels and MIR122 levels in the fetal liver, and it exerts antioxidant and anti-inflammatory effects in the placenta. Okay. Okay, with this and other studies I don't have time to show you today, we initiated a translational study under the hypothesis that an adverse intrauterine environment is ameliorated with diets enriched in olive oil in women with GDM. And for this purpose, we recruited 50 healthy and GDM women at the Hospital Pirovano in Buenos Aires between weeks 24 and 28 weeks of pregnancy. Women with obesity and other pathologies were excluded, and the control and GDM groups, the standard diet was recommended. And in a group of GDM women, this standard diet was enriched in 3 tablespoons of crude extra virgin olive oil daily. And in the placenta, we found again that PPAR gamma and PPAR alpha were reduced. There were no changes in the levels of PPAR gamma with the diet enriched in extra virgin olive oil, but PPAR alpha levels were upregulated. We studied microRNAs that regulate PPARs, and MIR138 is a negative regulator of PPAR gamma in different tissues, and we found no changes in the levels of these microRNA in the placentas in the evaluated groups. But MIR519G, a negative regulator of PPAR alpha, was found increased in the GDM placentas, an alteration probably associated with the reduced levels of PPAR alpha, and there were reduced levels of MIR518G when the GDM patients were treated with a diet enriched in extra virgin olive oil, together with the prevention of the altered levels of PPAR alpha. We also found increased inflammatory markers in the GDM placentas, interleukin-1 beta and TNF alpha, which were prevented by the maternal diet enriched in extra virgin olive oil. And we studied the activity of matrix metalloproteinases. These proteolytic enzymes highly needed during placental development and remodeling, but a marker of a pro-inflammatory state when are overproduced and overactivated. And we found overactivation of matrix metalloproteinase 9 in the placentas of the GDM patients, and alteration prevented by the maternal diet enriched in extra virgin olive oil. In maternal blood, there were no changes in matrix metalloproteinases activity at enrollment, but at term, there were an increased activity of matrix metalloproteinase 9 prevented by the maternal diet enriched in olive oil. And I think this is important, as this may be a possible circulating marker of the effectiveness of a diet enriched in olive oil in GDM patients. And we also found changes over activity of matrix metalloproteinase 2 and 9 in the cord blood in the GDM from GDM patients, and alteration prevented by the maternal diet enriched in olive oil. And this probably reflects that the benefits of the extra virgin olive oil supplementary diet reach the fetal compartment. As general conclusions and take-home messages, we believe that experimental models of diabetes and pregnancy are useful to address diabetes-induced fetoplacental impairments and programming of offspring diseases, that the intrauterine proxy and pro-inflammatory environment and metabolic alterations are likely related to impaired PPAR pathways, that diets enriching extra virgin olive oil have beneficial effects in fetuses, placentas, and offspring. And our translational studies suggest that the dietary supplementation with extra virgin olive oil has beneficial effects in GDM patients. And finally, all my thanks to the members of the Laboratory of Reproduction and Metabolism in Buenos Aires, Argentina, our international collaborators, collaboration with the Hospital Pirovano, and to the members of the OLIDIAC project, which is a project that is running to address the effects of extra virgin olive oil in a larger number of patients. This is a multi-center study. And thank you for your attention.
Thank you very much. Thank you very much, Alicia. Very nice talk and lots of data in here. Excellent. We have one or two questions on the chat, but I would like to ask the first question is how do you, what is your guess about the molecules involved in the effect of extra virgin olive oil? I mean, is there any candidate for the effects that you are seeing?
I believe it's a combination. There are monounsaturated fatty acids, polyunsaturated fatty acids, their capacity to activate PPARs and other receptors, and polyphenols also are likely to play a role. So I think it's a combination. I think that it's difficult to separate. Probably minor effects are held from the different components. Probably polyphenols are related to the antioxidant effects, but also the activation of PPARs. And those effects related to lipid metabolism are probably mostly related to PPARs activation.
Okay, thank you. Okay, here we have one from Ana Elias Lopez. Excellent presentation. Do you know a model of gestational diabetes generated by an endocrine disruptor like BPA?
I don't know. I know that there are models generated by different testosterone administration in neonates, different models that generate programming of type 2 diabetes mostly, but probably then there are also models of GDM generated by intrauterine nutrient restriction. So if you can generate a model of GDM, it's something in a previous step in the time that will start the type 2 diabetes. It's in patients, it's the same. We all know that GDM is a previous step of a possibility to have GDM in the life of this patient.
Okay, Alvaro Santana is saying, thank you, Dr. Jauerbaum. Really interested result for comparison with ones we have in our group. I will like question one. Why did you decide to use 6% of EVO enriched diet and not lower or higher? And the second one is the same we were discussing before. Did you try to isolate any specific component from the extra virgin olive oil, which could be related with the PPR regulation?
Yes, we use this 6% olive oil addition to give half of the lipid content of the diet provided by the diet. So the rat usually has 11% of lipid content in the diet, and then we try this half part so that make a diet that may be translatable then to patients. And indeed, the same we did with the patients. It's about half of the content of the diet, lipid content of the diet in humans is higher. The recommendations for lipids, but half of this is provided by the diet enriched in olive oil. So we think that is a possible diet to be done.
Yeah. Another question from Grace Chung, or Shang, sorry if I don't pronounce correctly. It's a would the timing of the extra virgin oil enrichment in the human study make a difference? For example, enriching their diet earlier during pregnancy versus late in pregnancy, or versus the whole pregnancy period?
We haven't tested that in the patients. It's very difficult to make this study in patients. So we enrolled women at the week of GDM diagnosis and not earlier. On the other hand, we didn't test in the animals only the diet from the effects on the last week of pregnancy. So it's something that I don't have the response, but it's probably make differences.
And the last one is from Paola Valero. Thank you very much for your presentation. What will be the recommendation then for the consumption of olive oil in a pregnant woman?
We are now running a study in a bigger number of patients, and will give us more reliable results. Other thing I didn't have time to show you here is that this diet in the we're able to change and reduce markers of insulin resistance and to reduce triglycerides in blood. And also there was a tendency, and we will see it in a large number of patients, a tendency to reduce the insulin dose, which is something interesting. So I think we will have more responses in this after when this large study, but as you all know, a large study in patients is something quite difficult to finish. So we are in the middle of this.
Okay, right. Thank you very much. Thank you for your lecture and for the question to the people then. Thank you, Alicia, again.
Thank you.
Okay, I will introduce myself. I have to talk now, and well, I'm working at the Pontificia Universidad Católica de Chile in South America, and I'm interested for many years already in placenta and fetal vasculature, and I wanted to show you today data regarding diabetes, and particularly gestational diabetes, but trying to introduce a concept that we call gestational diabetes recently. So I will try to show you some information to convince you about this new metabolic condition. We all know, and we just heard about this, gestational diabetes is a syndrome, and it's difficult because it's carrying very many perinatal risks, as reported in the American Diabetes Association this year and last year, and also is linked to very different postnatal risk, like including in the mother and in the offspring. And we have hypertension, the well-known type 2 diabetes, dyslipidemia. And in the offspring, we have obesity, childhood obesity, which is a problem worldwide now, metabolic syndrome, of course, hyperinsulinemia. But one of the characteristics also reported in this review in 2020 by Professor Lucilla Poston in England and colleagues is the vascular dysfunction that you can see in the list here for women and also for the offspring. Vascular dysfunction can be then observed in gestational diabetes as an endothelial dysfunction, and regarding the original definition of endothelial dysfunction in the mother, in the fetus, and of course in the placenta. Normally, the endothelial function, normal function, is described. One of the aspects described is the uptake of this cationic amino acid arginine, and the metabolism of arginine by the nitric oxide synthesis, particularly the endothelial isoform in endothelial cells. And this link between arginine transport and activity of the nitric oxide synthase to generate nitric oxide and then cause vasodilation is the L-arginine nitric oxide signaling pathway. Arginine entry to the cells is mediated by membrane transporters of the system Y family or cationic amino acid transporters of relatively low affinity, and also another family of transporters of very high affinity, the Y plus L transporters, are responsible in endothelial cells, particularly from the human placenta, to incorporate arginine to the cells and offer the arginine to the metabolism by NOS to generate nitrogen. So any disruption of this mechanism in the endothelial cells is also related with endothelial dysfunction. We want to check whether umbilical vein endothelial cells from humans, from mothers with gestational diabetes mellitus, showed any alteration in the capacity of taking up arginine from the extracellular space. And what we found here was that the cells derived from gestational diabetes show higher L-arginine transport compared with cells from normal pregnancies. And this increase in L-arginine transport was associated with an increase in the protein abundance in the endothelial cells of this isoform of cationic amino acid transporters, the isoform 1, which is with a Km around 100 to 200 micromolar arginine, right? The concentration of the plasma of this amino acid in humans. Further, we look at of course, the ability of these cells to generate nitric oxide. And what we found here is an increase in the generation of nitric oxide, which seems to associate with the increase in L-arginine transport, as I just showed you in these cells from gestational diabetes. But other actors are involved in this phenomenon. And for example, we know that other vasodilators like adenosine are also involved in the regulation of endothelial cell function. And adenosine, acting on adenosine receptors, will modulate several signaling mechanisms, including the generation of nitric oxide. It is well known that adenosine increased the level of nitric oxide in several cell types, including the endothelial cell. And also, there is an increase in the cyclic AMP levels and other molecules important for regulation of nitric oxide synthase, like the protein kinase B or AKT. But the extracellular adenosine level is critical for the activation of adenosine receptors. And one of the mechanisms to maintain the proper level of adenosine extracellularly in the human umbilical vein endothelium is the ability of these cells to take up adenosine through the nucleoside transporters. And these nucleoside transporters are, when they are not working well or they're very low in expression, will result in accumulation of adenosine extracellularly, increasing the biological actions of adenosine in these cells. So we want to see whether these cells from gestational diabetes show any alteration in adenosine transport via one of these transporters, one of these nucleoside transporters called the human equilibrative nucleoside transporter isoform 1, which is the most abundant expressed protein in this QX from humans, I mean, umbilical vein endothelial cell from humans. And what we saw here is that the uptake of adenosine is reduced in gestational diabetes compared with cells from normal pregnancies. This reduction was also seen as a higher level of adenosine in the umbilical vein blood in gestational diabetes, as is shown in this second bar, black bar here, compared with normal pregnancies. This is only for the umbilical blood from the vein, but not from the arteries, suggesting that the change in adenosine concentration seen in the umbilical vein was perhaps due to some alteration in the placenta and not as a proper alteration in the human fetus for the handling of adenosine. And this is important because adenosine is able to vasodilate the human umbilical vein rings, as was first published in 2011 by Francisco Westermeyer, now in Austria, in fact. One of the explanations for this reduced uptake or transport of adenosine to the cells include, for example, a reduction in the number of membrane of transporters and the plasma membrane in these cells in gestational diabetes, as measured by specific binding of NBTI, a molecule with high affinity for the nucleoside transporters. And also, as you can see in the picture here, there is a distribution in the cells with large major distribution in the plasma membrane. But then when we look at the cells in gestational diabetes, these transporters as the plasma membrane migrate to intracellular spaces, forming clusters of transporters, reducing then the bioavailability or disponibility of these transporters at the plasma membrane of these cells. So that may also result in reduction of the adenosine uptake. If we look at the promoter activity of the gene coding for the equilibrated nucleoside transporter 1 protein, the SLC29A1 gene, and we measure the activity of the promoter, we can see that in gestational diabetic cells, there is a reduction in the activity of this promoter. It's apparently this reduction is due to an activation of some sequence located between 2154 and 1810 pair of bases from the transcriptional site from this gene in gestational diabetes. If we look at the effect of some molecules that might be involved in this reduction of the expression of this gene, this finding is complemented with others that I'm not showing here, shows that the reduction of the promoter activity for this gene in gestational diabetes was reversed when the cells were pre-incubated with nitroarginine ester, which is an inhibitor of nitric oxide synthase, suggesting that this reduction perhaps was due to overaccumulation of nitric oxide in these cells from gestational diabetes. Interestingly, if we check in the literature, the section of the promoter between 2000 around to the other section of the promoter that I just showed you, around 1800 pair of bases, we found several consensus sequence for different transcription factors. But one of these, which is indicated with the arrow here, is the CHOP-C-EBP alpha complex of transcription factor, which has a very specific sequence of in the promoter. And interestingly, this CHOP-C-EBP alpha has been shown in the literature in different cell types that is activated when the level of nitric oxide is increased. And remember that in gestational diabetes, we have more production of nitric oxide in these cells from the human umbilical vein. So knowing that this is, sorry, I cannot move here. Knowing that this transcription factor was perhaps involved because the elevated level of nitric oxide, we measured then the binding, the specific binding for CHOP into this region of the promoter of the gene, and we found that the binding or activity of CHOP was increased in gestational diabetes. This is a transcription factor that is repressing the expression of this SLC29A1 gene in these cells. So more activity, more binding means reduction of the expression of the gene. If we look at now the same experiment in cells that were knocked down for nitric oxide synthase, endothelial nitric oxide synthase, this increase in the binding or activity of CHOP was back to normal or even reduced a little bit more in cells that were lacking a normal expression of enos. So supporting the possibility that activation of CHOP was due to elevated synthesis of nitric oxide in these cells. As a summary on this part of the presentation, the elevated node seen in gestational diabetes might activate these transcription factors, and then acting on the promoter, reduce the expression of SLC29A1 for a lower or reduced expression of the protein that will then be lower at the plasma membrane, reducing the entry of adenosine via this protein in the cells. That might explain why we see a lower uptake and why we see accumulation of adenosine extracellularly in these cells. But adenosine is involved in several pathologies, as reported some years ago already by in a review by Professor Bertil Fredholm. And you can see that the colors here are the different receptors, and you can see that diabetes is also involved with some of the receptors, A1 or A2B receptors, for example, in humans, and other diseases like cancer, etc. Adenosine receptors are in QX and other cell types are 4 different types, A1, A2A, A3, and A2B. And the mechanisms associated with their activation are very much different, but some of the common molecules are mentioned here, and one of these are cyclo-AMP, nitroxide, AKT, and others. So we want to see in these cells where these receptors were involved. And what we found here is that in gestational diabetes, there is an increase in the expression of A1 adenosine receptors and a reduced expression of A2A adenosine receptors. And A2B and A3 were unaltered for these cells. So one of both of these receptors might be involved in the phenomenon we were seeing and the regulation of vascular tone. If we look at L-arginine transport now and express their uptake as a maximal transport capacity, which is the ratio between the Bmax and Km, we found that in gestational diabetes, with cells that are normally expressing A2A adenosine receptor, the uptake is increased in gestational diabetes compared with normal pregnancies. But when we do the knockdown for A2A adenosine receptors, the increase is reversed back to normal values of maximum transport capacity. This is also seen in a pharmacological approach using M241385, which is an antagonist for A2A adenosine receptor. And you can see that the increase of maximal transport capacity in GDM was reversed to normal also. So that involves A2A in the adenosine accumulation and action to activate arginine transport and nitric oxide synthesis in these cells in what we call some years ago already the ALANO pathway in HUVEX from gestational diabetes. ALANO meaning adenosine L-arginine nitric oxide signaling pathway in these cells. We now know that A2B adenosine receptors might be also involved, and in fact, A2B is kind of protecting the endothelial cells by reducing the elevated level of ROS or inflammation, that increasing even more the level of nitric oxide that you can see, or we can see in the cells in gestational diabetes. But gestational diabetes has been studied for many years in groups of patients. Women that, in fact, were not considered to be separated because of their pre-pregnancy conditions. And we wanted to highlight here in 2020 that gestational diabetes could be in lean women, or could be in women that show pre-gestational overweight or pre-gestational obesity. And we believe that these 3 different conditions are in fact different metabolic conditions. If we look at the woman showing pre-pregnancy obesity, here is just a table to show that the BMI was, for example, 32 or around in a group of women with pre-gestational maternal obesity compared with normal pregnancy, just to show you that obesity is a condition that will alter the endothelial function in human pregnancy. And you can see that the PGMO, women with maternal obesity, the response to insulin of the human umbilical vein for dilation in normal pre-pregnancy weight of the woman is of these kinetics. However, when the umbilical vein comes from gestational maternal obesity, there is no response to insulin. And this may be due to reduced generation of nitric oxide in these women with pre-gestational obesity. However, the arginine transport in this case was not reduced as was expected because the reduction of nitric oxide, but was increased, and this is something that we are still studying. These cells also show in the plasma reticulum stress, which is an indicator of malfunction in these cells, of course. And one of the molecules of canonical signaling sequences is this one here in the title, the PERK EIF2 alpha CHOP and TRV3 sequence, which is shown here in the graph at the bottom, that there's an increase in the activation of PERK, an increase and activation of EIF2 alpha, increased level and perhaps activity, as I just showed you, in gestational diabetes of CHOP and also TRB3, including then or suggesting that this endoplasmic reticular stress via PERC is also activated. If we look at the ATF6, another sensor of endoplasmic reticular stress in the cells, it's also shown that the migration of the ATF6 in cells from normal pregnancy to the nucleus is like this, and in this figure here. And if we look at cells from pre-gestational maternal obesity, the migration to the nucleus is much increased. This is quantified here. This is the migration in normal weight pre-pregnancy, and this is in obesity pre-pregnancy. So implicating ATF6 in these alterations by obesity. Also, and I'm going to this quickly now, the JNK1 activation is also activated in cells from pre-gestational maternal obesity. And this is very important because JNK1 elevated and this activity is also involved in insulin resistance in many tissues, including the endothelial cells now. So obesity, pre-gestational obesity by itself is increasing or altering several mechanisms that will perhaps result in alterations in nitric oxide synthase and arginine transport in these cells. But in Chile, our reality, as it is also in the world, the prevalence of gestational diabetes is increasing from 2004 to 2020, from 7 to 20% gestational diabetes, and 60 to 80% of the, or 70% of the women are with obesity, pre-pregnancy obesity. So this is important. And what we proposed recently was that maternal pre-pregnancy BMI is important in the woman for the potential alteration that may cause the declaration of gestational diabetes on the fetal placental endothelial dysfunction. And we can see at least 3 different groups of women: women with normal weight, pre-pregnancy normal weight, overweight, or obesity. And whether a woman with normal weight, pre-pregnancy normal weight, develop gestational diabetes. That is what we know as the classical GDM or GDM lean, and whether the woman was with obesity before pregnancy, we are already defining it as a gestational diabetes. And if we look at the markers that are already published in the literature, we can see that gestational diabetes shows different pattern of intensity of the alterations causing the endothelial function associated with gestational diabetes compared with GDM lean or classical. So this information is also associated in the literature with a generalized reduction in the anti-inflammatory response in endothelial cells in this condition of gestational diabetes. So it's in fact compared with normal or lean gestational diabetes or normal pregnancies. I wanted to show you just some very quickly to finish now some evidence of the difference between gestational diabetes and gestational diabetes. And here is gestational diabetes in the group. The pH, intracellular pH in Curex from normal pregnancy is around 7.1, 7.2. And the pH in the gestational diabetes is very much alkaline, 7.7, compared with normal pregnancies. And if we look at the capacity of these cells to recover after an acid pulse experimentally induced, we can see that the capacity of the cell from gestational diabetes is higher than what we see in normal cells, suggesting that the mechanisms of the plasma membrane to extrude protons and then regulate the intracellular pH is higher than in normal pregnancy. So some transporters there are working more than in normal cells. So we use cells from normal, overweight, or obese patients. And what we saw that the basal pH was increased in normal weight and overweight and obesity, that women that develop gestational diabetes compared with normal pregnancies. And even more important was that the velocity to recover from an acid pulse was also increased in these cells from gestational diabetes and the three groups, normal, overweight, and obesity. But, however, please notice that the capacity of recovering from a pH that was acid, experimentally induced acid, was even more than normal, but it was lower than the capacity of shown by the normal weight of overweight gestational diabetes patients. If we look at now what kind of transporter might be involved, we used hexamethylene amyloride, which is an inhibitor of a group of transporters known as sodium proton exchangers in these cells. And we use also soniporide, which is specific for one of these isoforms, the NHE1 isoform of this transporter. And you can see that in normal weight and overweight, the potential action of NHE1 isoform is full. I mean, it's responsible for the change in the velocity of the capacity of the cells to recover the pH, and also in overweight, but not in obese women with pre-gestational obesity. Only half of the response is reversed, but other half is mediated by other transporters. So here is the scheme. In normal weight or overweight, NH1 may be involved in the intracellular alkalization of the endothelial cells, and in gestational diabetes may be NH1 plus other transporters also. If we look at nitric oxide synthase, it is reduced in obesity, as I showed you before, and is increased in gestational diabetes. But when these women are with pre-gestational maternal obesity plus GDM, now the synthesis of nitric oxide is increased very much compared with the other conditions. So it's showing again a different kind of metabolic condition. Looking at the adenosine transport, it happens exactly the same. There is a reduction in gestational diabetes and the normal weight, or a reduction in overweight, and a almost non-uptake of adenosine cells from pre-gestational maternal obesity, or what we call gestational diabetes. So what we know about gestational diabetes, for now, we know some information about adenosine transport, the regulation of intracellular pH in gestational diabetes involving the NH1 isoform, and some of the modification of nitric oxide associated with adjuvant transplant of the cells. Other factors, we're just thinking in what can be also another factor involved in this, and we are in collaboration with people in the Netherlands, looking at the effect of hydrogen sulfide as a gasotransmitter regulating nitric oxide also in gestational diabetes, and another transcription factor involved in the regulation of intracellular pH and nitric oxide is the activator protein 1 as a second chance for the regulation of nitric oxide and adenine and adenosine transport cells. And I want to finish now just to mention that these biochemical factors I showed you are involved in gestational diabetes and diabetes, but also there is an attempt now in the literature to identify factors before the declaration of the pathology, so to serve as a marker for the disease before this is identified. And here we have a record of glycemia using the continuous monitoring glucose measurement aggregated in 24 hours for 12 days or 14 days. And we can see that there is a nice distribution of the glycemia values during the day, night, or during the day, with a large variation also. But then Paola Valero published this just now, showed that the time in the range of acceptable values for glycemia might be reduced, and that is associated with some modifications, like, for example, microsomial or VN fetus enlarged for gestational age. All the excursion of glucose in these during these changes of glucose in the measurement we are doing are also associated with preeclampsia, insulin resistance, and others. The values that we normally use as a mean or standard deviation also associated with some of these alterations, and the index for glucose, low level or high levels, also associated with oxidative stress and inflammation. But none of them is mentioned in the literature for gestational diabetes. And Valero suggested that knowing some of these metrics isolated, we can identify in the continuous glucose monitoring system. I just show you 3 or 4, but here we have 25 or 26 different parameters. The mother that develops gestational diabetes will alter the placenta, and the fetus will have an adverse pregnancy outcome finally, causing with these kind of alterations. But if we can isolate one of these metrics early, before even pregnancy, perhaps we can do a kind of prevention for gestational diabetes and reducing the adverse pregnancy outcome, because that will allow the clinicians to have a good or better treatment of the patients, knowing what is the problem on the glycemia values and dynamics in these patients. So I wanted to finish with that, just saying that the mother is not a good idea to put all the women that are normal weight, overweight, obese as the same condition, because these 3 different conditions will alter the placenta differently, and the outcome with the newborn will be also different. Of course, avoiding obesity and overweight will improve this vicious cycle that we are all working in. So thank you very much to the people. Here we have some PhD students, Gonzalo, Paola, Marcelo, mainly, Adriana, that have been producing the data. Thank you very much for that, for your attention. Sorry about the time. Question if you have. It's difficult for me to do both, but I will start from the last one. Could be there from Florencia Savio. Could there be an interaction between the alanine pathway and PA3 kinase pathway? Thank you. Yes, we think it is. We think that there is an interaction. And when we look at the effect of insulin, which I didn't show here, PA3 kinase is much involved in the regulation of the beneficial effects of insulin, in fact, in gestational diabetes. So perhaps we can talk later about the mechanism. Ana Elias Lopez, sorry. Okay, I think we have those. Pablo Mendoza, thank you. I wanted to know if there is a proposed mechanism to explain the contribution of adenosine receptor A to A to B on nitrogen transport and neurogeneration. Is an effect on the translocation of the transporter? Yes, expression. Is cycling accumulation involved? Okay. Yes, thank you very much for the question. Yes, there is an interaction there. We know by the literature that in pathologies like gestational diabetes or type 2 diabetes mellitus, the time of residence of adenosine in A2A and receptors is increased largely 3 or 4 times. So that will, in fact, maintain a larger signaling in the cells for a longer time, activating many of these mechanisms I just showed you, including the nitric oxide synthase and the arginine transport in the cells. Cyclic AMP accumulation was measured, I didn't mention it, and looking at the cyclic AMP levels, we found an increase in gestational diabetes and also in response to adenosine receptors. That's why we didn't, in fact, measure the with much information the dedication. I mean, the A1 adenosine receptor, because it's down regulating the adenylyl cycles in the cell. So we went straight to A2A adenosine receptors in these cells. Thank you. If I have more questions, I will reply by the chat. So sorry about that. Thank you very much. And I have to move now to our next speaker. The next speaker is Professor Gernot Dessois. Yeah, he is on the screen. And Professor Dessois, we all know him very well for gestational diabetes, obesity, the concept of diabetes also, and all his contribution as a biochemistry in the field. He has received many prizes, of course. We know that he's recently awarded by the American Diabetic Association of a very important prize on the research and contributions in diabetes. So I don't think I need to introduce very much. We all know Miguel Lopez, and we are thank you for his contribution and teaching permanently in our meetings and congresses about diabetes. So thank you again.
So thank you very much, Luis, for this very kind introduction, and actually also flattering introduction. And thank you to everybody for staying here. I don't know what the time in the various regions of the world are. How do I get rid of these? Oh, I don't know. Okay, so you have heard already several times from the previous speakers that overgrowth is one of the consequences of maternal diabetes and also of maternal obesity. This is a fetus, or now a neonate, born to such a pregnancy. And the usual terms that are used to characterize this neonate is it's born large for gestational age, or LGA, or born macrosomic. However, these are actually birth weight centered classifications, and they do not tell us which component of the body or which compartment of the body is actually overgrown. So if you just use a simple two-compartment model based on and comprising fat and fat-free tissue, then it has been shown numerous times now that there is an increase in the fat compartment and no change in the lean body compartment. And here's just data from Pat Catalana's group in which he clearly demonstrates that there is an increase in the body fat, not only in offspring born large for gestational age, but also in offspring born with appropriate for gestational age. And this is important because it has enormous implications for public health. So usually we focus, or the MDs or public health activities focus on the offspring born LGA or macrosomic, but there is also important to realize that the offspring born with an inconspicuous birth weight appropriate for gestational age, if they have an increase in body fat, are at higher risk for developing childhood obesity. So this increase in the body fat has now been demonstrated in offspring born to type 1, type 2 diabetes, to gestational diabetes, and to obese women. So this would suggest that there is a common pathway underlying the increase in the fetal fat deposition. And this is indeed the case. This pathway has been described long ago, and we have modified it, expanded it a bit. It starts with maternal hyperglycemia, which is common to all these types of diabetes, and when women are obese and unhealthy obese, I have to say, because there is also a healthy obese phenotype. And this maternal hyperglycemia then results in fetal hyperglycemia, fetal hyperinsulinemia. And both glucose and insulin then lead to an increase in the formation of fatty acids in the fetal liver, resulting in triglyceride formation that is enhanced, and the triglycerides are then packaged in lipoproteins, foremost but not exclusively in HDL. And HDL then reaches the white adipose tissue, and with the support and stimulatory action of insulin, the triglycerides are hydrolyzed by lipoprotein lipase on the surface of the white adipose tissue, and the released fatty acids are taken up by the adipocytes to form triglycerides. And also fatty acids can be synthesized de novo in the adipose tissue, in the adipocytes, in the white adipocytes, out of glucose as a precursor under the stimulatory activity of insulin. So it's really the glucose-insulin axis that drives the increase in fat deposition. And that raises the question, what is the role of the placenta, if any, is in this pathway? Or more specifically, the question comes up whether glucose transfer across the placenta is increased in these conditions that lead to an increase in fetal fat deposition. So we know that the transfer of glucose across the human term placenta is rapid, it's efficient. The responsible glucose transfers have been identified, and the most prominent one, GLUT1, is a sodium-independent glucose transporter, and that is present in all cell types in the placenta. It is regulatable by a wide range of molecules, including hormones, metabolites, cytokines, oxygen, growth factors, and also these glycerides, sulfonylurea, that is often used in the United States, particularly to treat women with GDM. So with this in mind, it comes as no surprise that GLUT1 expression, protein levels, activity are altered in diabetes of all kinds, and also altered in fetal growth restriction. So that these alterations have long suggested to us in the field, to everybody who was working in the field, that the maternal fetal glucose transport is increased in conditions of maternal diabetes. And in fact, we were all surprised when the results were published by Neil Good's group from Melbourne 20 years ago, in which he used the ex vivo placental perfusion model to measure maternal fetal transfer of glucose. And what they found there was that the transfer was actually unchanged. And here you see the data that have already multiplied by the total placental weight, because in that publication they did have some differences in placental weight in the various groups. So I multiplied it with the total weight. So this would suggest that there is, even with the bigger placentas that they had in the GDM groups, there is no change in glucose transfer. And just parenthetically remarked, also there is no change in fetal growth restriction, at least not when glucose transfer was measured by this ex vivo placental perfusion model. And the reduced transfer of glucose in FGR in fetal growth restriction is mostly accounted for by the small size of the placenta. So it seems that transplacental glucose flux, at least at the end of gestation, is not profoundly determined at the level of the placenta. So that leaves the maternal fetal glucose concentration gradient as the main dictator of glucose flux. And that has important implications, because the glucose gradient is not only determined at the level of maternal glucose. So with hyperglycemia, you have a steeper gradient, but it's also determined at the side of the fetus. Fetal glucose, as I already explained, results in fetal hyperinsulinemia, and the hyperinsulinemia then facilitates extraction of glucose into peripheral tissues of the fetus. So whenever there is hyperinsulinemia, then there is a transient reduction in glucose in the fetal circulation, and that then steepens the concentration gradient. So with hyperinsulinemia, the glucose flux is actually independent of the maternal, or becomes not only dependent on maternal glucose concentration, but also is enhanced. This seems to reflect a situation in which the fetus actually pulls glucose from the mother. And we have, or actually Chris Nolan has given this phenomenon the name fetal glucose steal phenomenon. So fetal hyperinsulinemia is really something that has to be avoided, or at least its onset has to be postponed when we want to prevent, or when we aim for preventing the increased formation and deposition of fat. So how about the fatty acids? The second component that is important when we speak about fetal fat accumulation. So fatty acids can cross the placenta, but the transfer characteristics that have been worked on and shown decades ago find that the transfer is very slow. It's inefficient. Only between 2 and 4% of fatty acids in the maternal circulation transfer to the fetus. The transfer depends on the chain length of the fatty acids. And then because of this inefficient transfer, only a small contribution, and was calculated to be about in the range of 20% of maternal fatty acids contribute to neonatal fat in a normal pregnancy. So the question arises then, what happens when there is metabolic disturbance in the mother? Birgit Hirschmuggel in our lab used the ex vivo placental perfusion model and to measure fatty acid transfer from the mother to the fetus, and compared the transfer between lean and obese women. And what she found was a significant change in the transfer of DHA, docosahexaenoic acid, which is important for fetal brain development and formation of the retina. She didn't see any significant difference in other fatty acids that she has used, including palmitic acid, which is the most prominent fatty acid in neonatal fat depots. But as you can see here, there is a slight increase in all these fatty acids transferred to the fetus when the mothers were obese. It just didn't reach significance. So we cannot exclude that there is a slight increase in fatty acids that get across the placenta in conditions of maternal obesity. But nevertheless, it seems that there is not much of the placenta that contributes directly to the enhanced fat deposition in the neonates that are born to pregnancies with all kinds of diabetes and or to obese mothers, at least at the end of pregnancy. Now we are seeing a lot of changes in the placenta associated with diabetes and obesity, and Alicia and Luis have just characterized some of them. And in my understanding, some of these changes that we're seeing are actually adaptive responses to the altered maternal environment and also to the altered fetal environment in order to protect the fetus. And I will just give you two examples. These adaptive responses are important because the hyperglycemia and hyperinsulinemia stimulate the aerobic metabolism in the fetus, particularly in the liver, and that requires more oxygen. And this is initially dealt with by an increase in the oxygen extraction. But if the enhanced aerobic metabolism and enhanced oxygen demand is of longer duration, then there is also an increase in the oxygen supply to the fetus. And this is brought about by two different mechanisms. There is an increase in the oxygen transfer, and also the blood supply to the liver is enhanced. So one of these protective mechanisms against fetal hypoxia is an enlargement in the placental vascularization. There is indeed evidence that the fetus can be hypoxic in gestational diabetes, and higher erythropoietin levels were found in the umbilical cord blood, or an increase in the hematocrit because of more red blood cells. And the hypervascularization of the placenta has been demonstrated several times, or umpteen times I can say, and the capillary surface area in diabetes can be increased up to 50%. And because of the elongation of the capillaries, the maternal fetal diffusion distance is reduced, and the ultimate aim is therefore that the oxygen transfer across the placenta is facilitated. We have identified a couple of pathways, all stimulated by insulin, that facilitate or stimulate the angiogenesis that leads to the hypervascularization. The first one is through the activation of the AKT. Matrix metalloproteinase 14 is increased and its activity is enhanced, and MMP14 is one of the important players in angiogenesis because it facilitates the degradation of extracellular matrix. And of course, when the vessels want to or need to be enhanced or enlarged, extended is the right word, sorry, then you have to degrade extracellular matrix to make space for the growing vessels. AKT activation, as we have just heard, also leads to an activation of enos and nitric oxide formation. And one of the consequences of nitric oxide formation is also the formation of tubes, vascular tubes. And through activation of BF3 kinase, insulin can also activate the small GTPase RAC1 that we have demonstrated being involved in stabilization of the actin cytoskeleton. That is important again for lamellopodia formation and cell migration. So there's a couple of processes that we have identified and signaling pathways delineated that are activated by insulin, and that ultimately can contribute to increased angiogenesis, which will ultimately lead to the increase in the vascularization. I also said that there is one other mechanism important, and that is to ensure that there is adequate blood flow to the fetus. What the fetus does not want to have in that situation is the formation of any atherosclerotic plugs that would impede the blood flow. So one of the adaptive responses that I want to show and share with you that we have found is the protection against fatty streak formation. It is very interesting that in conditions such as type 1 diabetes or hyperlipidemia, there is acute atherosis on the maternal side in spiral arteries, or foam cells have been seen in the intervillar space. And also in type 1 diabetes, some increase in lipid accumulation have been found in several fetal aortas. But no such lesions have ever been seen or reported in the literature in placental vessels. And that suggested to us that there must be very efficient mechanisms operating that prevent these formation, and particularly operating to facilitate the release of and removal of cholesterol from the endothelial cells and from the fetal circulation, respectively. And we have identified some of these mechanisms, ApoE and ApoA1, synthesized by the fetal liver, can efficiently and effectively pick up cholesterol that is released from the fetal endothelial cells, fetal placental endothelial cells, through the efflux transporter ABCA1. That results then in the formation of pre-beta HDL, which again can pick up cholesterol from the endothelium that is then released through the ABCG1 transporter to form HDL3. And HDL3 can then be modified by phospholipid transfer protein to lead to pre-beta HDL again that can pick up more cholesterol, and the HDL3 is then modified to cholesterol-rich HDL2 that is taken up by the liver and sequestered there. In diabetes, in gestational diabetes, we found that the endothelial cells produce more cholesterol, and this more cholesterol is then released through ABCA1, and there is more ABCA1 in the endothelial cells in gestational diabetes, and there is also more of cholesterol efflux to pre-beta HDL. And also ABCG1, the second transporter, or efflux transporter, is upregulated in GDM, and also the efflux of cholesterol from the endothelial cells to HDL3 is increased. We also found that phospholipid transfer protein is increased, and there is also more HDL2 in the fetal placental circulation. under conditions of GDM. So there is, in summary, there's at least two mechanisms that ensure that there is oxygen supplied to the liver and that there is adequate oxygenation of the fetus in conditions of increased oxygen demand. So their increase in vascularization of the placenta facilitates the oxygen transfer under condition of metabolically induced hypoxia. We have seen that there is an increase in the cholesterol release at various levels. The transporters are upregulated, efflux is upregulated, and also the phospholipid transfer protein is upregulated. And these adaptations are in part regulated by insulin in the fetal circulation. And as you see here, there's, and I've shown in one of the earlier slides, that there is hyperinsulinemia. So this hyperinsulinemia helps the placenta to facilitate the adaptation to fetal needs in these conditions. So the take-home message is that the fetal phenotype in diabetes is often characterized by increased adiposity. And the important information is really that this is independent of birth weight. So I would really ask you to keep this in mind. The fetal hyperinsulinemia, I think, is the main driver of fetal fat deposition. And the relative contribution of fetal glucose-derived fatty acids or maternally derived fatty acids to fetal fat formation and deposition is unknown, still unknown, and I think it will be enormously difficult to tease out which one is more important, and the proportion then, of course. And the fetal insulin not only stimulates oxidative metabolism that then entails enhanced oxygen demand, but at the same time, it also facilitates adaptive responses of the placenta to cope with this enhanced oxygen demand. So this is to say thank you to a number of people who have contributed to some aspects that I have shown today. And I thank you for listening, and I'm hoping for a lively discussion. Thank you.
Thank you, Gernot. Very nice. Thank you very much. We have some questions. We are tight with the time. So I have a question from Paola Casanello with presentation, Gernot. I was wondering if fetal hypoxia in GDM is really an oxygen transfer problem or an increased oxygen consumption by the fetuses.
Well, you hear me say, thank you, Paola. There is a increased oxygen consumption, oxygen need, oxygen consumption. And of course, the fetus tries to make more oxygen available. And there is, as I said, a couple of adaptive mechanisms. One is, as I said, the increase in the vascularization of the placenta. The other one is preventing that there is an obstruction to blood flow. And then there is, of course, all kinds of changes in the liver itself, and the liver perfusion is increased, and ductus venosus shunting to the heart and to the brain is a bit reduced in conditions of GDM. Well, this, in fact, has been demonstrated in type 1 diabetes, but the principal mechanisms are certainly also operating in GDM. It's just the difference between type 1 and GDM on the fetal side is just the extent of the hyperglycemia and then the extent of the hyperinsulinemia. So I think the mechanisms are all the same. The degree of the responses really depend on the level of hyperinsulinemia and or hyperglycemia.
Good, thank you. There's another one from Ashley Othman. Dr. Dessois, is there any information about fetal LDL levels in GDM?
Yes, there is information. There is a couple of papers by a group from Merzuk is the author, but I can't recall the data in the moment. Sorry, but if you send me an email, I can give you the literature.
Right. So Ashley, please email Professor Dessoin. Then we have Valentina Emily Vera. Thank you so much for the presentation about the length of the chain of the fatty acids. Is there any recommendation or suggestion to promote health during pregnancy?
Well, this would fill other hours of presentations. Can you be a bit more specific about what you mean in terms of recommendation? There's all kinds of lifestyle recommendations, physical activity, diet. We have heard from Alicia about dietary interventions. The question is really what you're ultimately aiming for. A healthy pregnancy with a healthy fetus should actually just aim to you should just aim to I think just use yeah, of course you should enrich docosahexaenoic acid in your diet. I think that is important, particularly when you have any form of diabetes or probably also obesity. There is, of course, the folate supplementation and all kinds of other recommendations. They differ from country to country and from organization to organization. So I without any more specific question, I can't give you a better answer. Sorry.
Yeah, it's all right. Perhaps Valentina could write to you later on, and then you can get a discussion on that. And the final question is from Paula Valero. It's a long one. It said, under hypoxic conditions, ROS is increased and affects the functioning of mitochondria and nitric oxide bioavailability. Is it possible that the increased angiogenesis in GDM is affected by ROS, and also whether oxygen transfer is still defective despite the increased angiogenesis favored by insulin?
Well, theoretically, I think it can be. The scenario that you sketched here may well be operating. I think it will be enormously difficult to really tease out what is what. Ultimately, I think there is instances in which, as shown by the increase in the erythropoietin level in GDM, there is instances where obviously the oxygen supply is not sufficient enough to prevent the increase in the erythropoietin. But we must not forget that an increase in erythropoietin at the very end of gestation in the cord blood will only tell you something about the oxygenation of the fetus in the past 12 or 16 hours. So we do not really have enough information or adequate information about the degree of oxygenation of the fetus during pregnancy, and we can only measure after delivery all kinds of parameters, including lactic acid, pH, and PO2, etc. So all kinds of mechanisms are certainly in play, and I cannot rule out any of these. We just see the net effect.
Yes, okay. Right, thank you very much, Gernot. Very nice discussion and presentation, of course, as always, and also Alicia Jauerbaum from Argentina. And all the people in the audience, they keep asking now, so please contact the presenters and they will give an answer to your questions. Thank you very much for that. And I will see you all next Friday at 2 p.m. GMT, all right? We have a second session on diabetes with people from Germany, James Kroll, from Brazil, Maril Sarruch, and from Canada, Professor David here. So thank you very much. Thank you again, and
thank you. Thank you, Luis and Alicia. And thank you to everybody.
Goodbye to everyone.
Bye-bye.
Enjoy the day. Bye.